Thank you. Yes, thanks everybody for joining today. In order to start on time and honor the time everybody who came early, I would like to introduce our very own scientist here at UTRGV, Dr. Alan Francis. He's an assistant professor at the Institute of Neuroscience here at UTRGV. Before coming here to UTRGV, he worked as an instructor of psychiatry at Harvard, the Mass General Hospital. He works on neuromodulation and neuroimaging in neuropsychiatric disorders and Alzheimer's. So he's interested in understanding psychiatric disorders like ADHD and comorbid with substance abuse disorders and using cognitive neuroscience methodologies. I think, well, Alan is also very versatile, and he has been working a lot on neurostimulation using TMS and other techniques. I would like to, well, Alan has been all around the world. He's has been visiting fellow in Oxford, a Master of Philosophy in the University of London. Also, his PhD was from Germany. So yeah, it's a very interesting personality. So let's welcome Alan Francis to our series of seminars. Really hope that you will enjoy his seminar. Thank you. All yours, Alan.
Thank you so much, Nay. Thank you for that wonderful introduction. It's always been a pleasure to work with you. And I also thank Naira for organizing this. This is a really good way of, sharing the research that each one of us does and sort of getting feedback from our colleagues and friends and students, and also showcasing our research for students so that if your students are interested in this form of research, they can actually come and, knock on our door and sort of join us. So without much ado, let me see if I can share. Let's see, let's share. Wait, just a moment. Okay. Okay, let me see if I can share this. Oh, yeah. Let me get that nice and big. Okay. Um, Hello, everybody. How's everyone doing? Good. So, my talk for this afternoon is going to be on the addiction connectome. We're going to look at multimodals, 7 Tesla imaging in cannabis, tobacco, and alcohol. Okay. Just a second. No little issues. Okay. Moving forward, I have no disclosures, financial disclosures that sort of compete with my research at UTRGV. And I have to declare that before every talk. So the outline for my talk, let me get this over here so it's easier that way. One second. Okay. The outline for my talk, just a moment, I'm just doing a bit of gardening here. Okay, that's great. Yeah, that's great. The outline for my talk includes, we're going to discuss what exactly is the addiction connectome. Second, we're going to discuss 7 Tesla scans and multimodal imaging. We're then going to move into tobacco studies, how 7 Tesla scans and multimodal neuroimaging can be used in tobacco studies. We'll then move into cannabis studies, and then we're going to look at alcohol studies. And then we're going to look at something called as knowledge gleanings. Why do we do this? I mean, what the heck's important about this? Why do we, what are the nuggets of knowledge and wisdom do we get from this? And then we're going to go into conclusions. What do we conclude from this? What are the commonalities between these three addictive disorders and what separates them? And finally, we're going to look at future directions. Where do we go from here? So, let's look at the addiction connectome. What the heck's the addiction connectome? The connectome, this picture from my colleagues at MIT, actually it captures it very beautifully. As you can see, there are color-coded balls. I'm sorry. I'm sorry. A little bit of. There are, when I move my cursor, it keeps moving. There are actually color-coded, there are color-coded balls here. The color-coded balls, purple balls, correlate. In other words, synchronously oscillate with other regions that are purple balls. Am I making sense to you? So, if a group of neurons in one particular area are synchronously active with another group of neurons in a different region, they are supposed to be functionally connected. It's on the basis of a Hebbian rule. Donald Hebb actually talked about the Hebbian rule. Neurons that fire together wire together. That's the fundamental Hebbian rule. So, in other words, when you look at these balls, they actually are interconnected in some manner. So you have these purple balls, you have red balls, you have yellow balls. So, in other words, our entire brain is connected, functionally connected, and they do their connectivity activity and they fire in response to specific tasks. And we are going to look at this is the idea of the general connectome. Now, we are going to look at the addiction connectome. The addiction connectome is defined as a network of brain regions and neural circuitry involved in the development and maintenance of addiction. Okay? Addiction involves a complex reorganization of interconnected neural systems, including those related to reward, motivation, decision making, and impulse control. These four cognitive characteristics are very symbolic of addiction. Let me just sort of rephrase that. In addiction, these four cognitive characteristics go awry, and that's why this whole aspect of addiction happens. Understanding addiction. Let me push it somewhere. Understand the addiction connectome helps researchers to identify targets for therapeutic interventions aimed at. I can't see what's this happening here. One second. Let's dismiss that. Okay. I'll just bring this over here. I have to do some gardening. My whole screen is kind of full here. Sorry about that. Okay. Uh, Understanding the addiction connectome helps research identify targets for therapeutic intervention aimed at disrupting the maladaptive brain circuits involved in substance use. Okay, so let's move forward with that. Okay, the next aspect I'm going to talk about is 7 Tesla scans. 7 Tesla scans, as you can see here in this picture, on your left, you have a 3 Tesla scan, and on the right, you have a 7 Tesla scan. What becomes immediately apparent to you is the amount of clarity you can actually see here. You can look at regions that actually show you, like, you can actually see a white matter, gray matter, CSF, and the pile surface, the different meninges, all of these are very, very, very clear. And that's why 7 Tesla scans offer a much better terra firma onto which we can do research. Okay, so let's look at why 7 Tesla scans are so important. The 7 Tesla advantages are as follows. Higher signal to noise ratio, that is, more signal, less noise. In radiological parlance, if there's too much noise, it obfuscates the signal. So we need more signal to noise to be able to sort of make a clear um, uh, to have a clear understanding of what's going on. So it provides a clearer, more detailed image. It 7 Tesla scans also provide for improved spatial resolution. It enables the visualization of regions called the hippocampal subfields, which are really, really tiny regions, but they are profoundly affected in diseases like Alzheimer's and alcoholism, and even neuropsychiatric disorders like schizophrenia, bipolar disorder. The hippocampal subfields play a very important role in all of these diseases. So, sort of viewing them is extremely important. 7 Tesla scans also offer better tissue contrast. It enhances gray and white matter differentiation and also susceptibility effects. It also, 7 Tesla scans also provide for enhanced functional imaging. And this is very, very attractive for someone who's involved in neuroimaging. When you see a 7 Tesla's uh, fMRI scan, it's really beautiful to look at. And you're able to sort of you're able to provide a much better understanding of what hypothesis you are actually testing. It offers 7 Tesla scans offer superior spectroscopy because spectroscopy is now used in psychiatric disorders, addiction as well, and neurodegenerative disorders as well. In spectroscopy, you can actually see the different metabolites, but some key um, metabolites like GABA, gamma aminobutyric acid, and glutathione. These are all, unless you have a very high a very high magnet strength, you're not able to see the GABA signal, the glutathione signal. And understanding how glutathione works in psychiatric disorders is a very important thing. 7 Tesla scans also provide for detection of small pathologies. It's ideal for identifying microbleeds, ion deposits, and subtle lesions. And finally, it's used for advanced neurological insights, facilitates research and diagnosis in epilepsy, Alzheimer's disease, um, and a lot. As an example, hippocampal abnormalities, if you look at on your right, you have a 3 Tesla T2 weighted scan, which, sorry, one moment. And on your left, you have a 7 Tesla T2 weighted scan. And actually, you can see a lesion at the hippocampal level, where the hippocampus is. And likewise, when you look at a 3 Tesla flare, which is a fluid attenuated inverse recovery scan. It's a very important T2 scan, which has its own it has its own. We use those scans for a particular purpose. A 7 Tesla scan looks very much more full of clarity in comparison with a 3 Tesla flare. So you can actually see the lesion here. Okay. Now that we have the basic groundwork, let's move into multimodal neuroimaging. Multimodal neuroimaging principally consists of looking at structure and structural connectivity. We also look in the same sample for functional connectivity, which is principally resting state fMRI. And we also look at diffusion tensor imaging and fiber tracking. And if possible, look at the autopsy or postmortem imaging. So let me explain it a little bit more clearly. So for a given group of subjects, if we look at, one second, if we look at structure, function, structural connectivity using DTI and PMI, we get a much more clearer understanding of what's going on with that group of subjects. Okay, moving on. Let's now look at nicotine uh, at 7 Tesla. One of the papers that we published in June, this was from my colleagues at Sophie is from France. Dr. Gabrieli is, I mean, she still is at MIT, and Dr. Campradon is at Massachusetts General Hospital. We published a paper that used multimodal 7T imaging, and we found that it reveals enhanced functional coupling between the salience and frontoparietal networks in young adult tobacco cigarette smokers. And this was published in Brain Imaging and Behavior in June. Essentially, in that, the study design involved, um, we basically were asking one principal question. Does smoking tobacco cigarettes, that is 20 cigarettes per day, affect intertemporal decision making as measured by a delay discounting task? If so, what are the neurobiological correlates of these cognitive deficits? So we used a stepwise neuroimaging design. We used four in vivo methods. We looked at functional connectivity in the tripartite network, and I'll explain to you what the tripartite network is. We looked at diffusion tensor imaging assessment, and I'm going to explain to you what FA, MDR, and ADR. And we did another imaging technique called whole brain NODI, which is neurite orientation, density, and dispersion imaging. And finally, we did cortical thickness of the nodes of the tripartite network. Okay, so that was our basic study design for this paper. So, explaining what a multimodal design comprises, we take a particular aspect of cognition. For example, in this case, intertemporal decision making or intertemporal choice. If we want to test intertemporal choice between a group of smokers and non smokers, we first take a cognitive task. And the cognitive task is, in this case, delay discounting task. So we first analyze what cognition is doing. And then we look at functional connectivity in the same group using 7 Tesla scans. And then we move on to diffusion tensor imaging analysis of the same group of people. And then we move on to NODDI, which is neurite dispersion, orientation, and density imaging. And then finally, we do cortical thickness analysis. So with this form of a multimodal design, we get a comprehensive understanding of really what is going on when someone smokes 20 cigarettes a day. Okay, let's move on. So where did this data come from? The data came from the Human Connectome project, which is at Washington University and University of Minnesota Consortium. 44 subjects were chosen from a total of 184 7 Tesla scans. The inclusion in the smoking groups was to smoke 20 cigarettes a day. Um, the smoking frequency was quantified using the semi-structured assessment for the genetics of alcoholism, which is called SAGA. It's a standardized test. We also use the Fagerstrom test for nicotine dependence, the FTND, and this test assesses the intensity of physical addiction to nicotine. So essentially, these were the this was our our repository of data. The hypothesis that we were actually testing were these. In cognition, we tested the hypothesis that smokers would score significantly lower on the delay discounting task, reflective of higher delay discounting and lower response inhibition. In functional connectivity, our hypothesis was the within network interactions of the frontoparietal network and the between network interactions of the salience network and the frontoparietal network would be altered in smokers. Now, I'm going to have one slide that explains what the salience network is, what the frontoparietal network is. So I'm getting to actually to that point. Um, the hypothesis regarding DTI was smokers would have reduced FA, which is fractional anisotropy, and I'll talk about that as well, in the regions associated with the salience network and the frontoparietal network. In other words, we were looking. One moment, my light's gone off. We were looking at functional connectivity on the top, and we're looking at the structural connections at the bottom. We're looking at both in a given group of people who smoked. Next, we looked at neurite orientation and diffusion density, dispersion density index. Smoking would adversely impact neurite density and orientation dispersion, as shown by the parameter maps. Okay, and then finally, we looked at the cortical thickness of the concomitant regions of the two resting state networks in smokers. So, what is the tripartite network? The tripartite network is a group of three networks. As the word tri represents, it's three different networks in the brain. And as the word network implies, it's not a single region, it's a group of regions in the brain. So, the salience network is a network that gets activated when when something that is salient to you, something that's important to you, is in front of you. For example, if you are thinking about something, you're thinking about when you want to have like lunch, the salience network shifts towards that. If you are doing a task, you're doing some mathematical calculations which needs absolute cognition, the salience network moves towards that. So the salience network is harbored in the anterior cingulate cortex and the left insula and the right insula. So these are the neurobiological basis of the salience network. The salience network, its principal job is a dynamic switching, and this happens automatically. Dynamic switching between the default mode network, which is harbored in the medial prefrontal cortex up here, and the posterior cingulate cortex at the posterior at the posterior part of the brain, and also the lateral parietal. Okay, so it switches between the default mode network and the central executive network, which again is harbored in the dorsolateral prefrontal cortex, both left and right, and the posterior parietal cortex over here. Okay, as you can see. And what do these two networks do? The default mode network handles self-referential action. That is, everything that's important to you. You need to go out and make it. You need to go and meet somebody. You need to make a phone call. You need to have a cigarette or you need to have a drink. All of that is handled by the default mode network. The central executive network, as you know, is a network that is active when you're doing a cognitive activity. It's externally directed action. So the salience network switches between the default mode network and the central executive network. And as I said before, it's not a single region, it's a network of regions. Now, the tripartite network acts in together. They act together. It's one thing. It either moves this way or it moves that way. Okay, so let's move on. So for this paper, which I just talked about, we did functional connectivity analysis. For that, we used the Con toolbox. And the actual design was this. We took smokers, those who smoke 20 cigarettes a day, and people who do not smoke at all. And then we put them into something called as component correction, which is a part of the Con toolbox. That is, when you collect functional images, what happens is um, several non-signal images will kind of masquerade as a signal. So you have a lot of noise coming in. So the ComCore does an excellent job of removing noise from the actual signal. It does a really outstanding job. Without component correction, you can have a lot of spurious findings in imaging. Imaging is it's a very delicate tool because sometimes noise can actually masquerade as a good finding. So, um, quality control in images is of absolute importance. You cannot begin to do statistical analysis unless you're absolutely sure that your images are actually telling you something good. So the component correction within the Con toolbox takes away spurious signal like noise and gives you the actual signal. And then we In this study, seeds were actually were placed in 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 four different regions. We put seeds in the right and left anterior insular cortex, the right and left, the dorsal anterior cingulate cortex, the right and left medial prefrontal cortex, and the right and left posterior parietal cortex. Now, each of these belongs to one aspect of the tripartite network. Um, AIC is within the salience network. The DACC is also salience network. MPFC is in the default mode network, and the posterior parietal cortex is within the frontoparietal network. And then we registered this with the Harvard Oxford Atlas. We did the registration with that. We also did whole brain DTI using FSL. FSL 5.0 track-based spatial statistics was done. For those of you who are interested in FSL and who are really interested, FSL 6.0 is out there. It's really, really advanced, and you can download that and kind of play around with it. It's really, really nice. So FSL has its own inbuilt toolbox to do whole brain DTI. And then we ran something called as randomize with 10,000 permutations to try. Essentially, what this does is that it gives you a coherent analysis of the uh, diffusion directions. And then we did stringent whole brain analysis with family-wise error-corrected P. And then once the analysis was done, we viewed it with FSL eyes, or otherwise known as, it's called fossil eyes. And again, the registration was done with the Harvard Oxford Atlas. We also did the uh, Johns Hopkins University White Matter Atlas and the Jülich Histological Atlas. Now, if you're thinking, okay, you have to open these atlases. Well, FSL has a particular menu. You can actually pull down the atlases from there. So when you pull down the atlas, you can actually see how the registration is actually done. Just one second. Let me just put this up here. It'll be easier then. Okay. 1 second. Okay. That's interesting. Can't see that. Okay. Um, just to talk about what diffusion tensor imaging is. Oops. Small types of gardening, sorry. So in diffusion imaging, essentially, I'm not going to give a long talk about what diffusion imaging is. It's basically taking a T1 scan, which is a basic structural scan, and also acquiring different directions. You get different directions in the multiples of 8. So it's 8 directions, 16 directions, 32 directions, 64 directions, and 128 directions. So then you actually you co-register those directions onto this T1 scan, and you are able to get a 4-dimensional scan from which you calculate the tensors. And the tensor is a vector quantity. In other words, it has magnitude and direction. So, and then you calculate fractional anisotropy. Fractional anisotropy is a um, a measure of how water diffuses across axons. It reveals to you the coherence of an axon. If there is an axonal problem, it will tell you there's an axonal problem. So it looks at how coherently water diffuses across axons. So it's called fractional anisotropy. And then you do what's called as fiber tracking. You actually look at a particular track and see how whether there's a problem in the track. And then finally, you have white matter delineation. So diffusion tensor imaging involves all of these. Now, if you're thinking, oh my God, that's so complicated, no? Usually programs do it for you. In other words, as long as you know what you're doing, the program will actually kind of lead you into it. The key thing is to be able to know what you're doing and why you're doing it. And the program will actually lead you through this. So essentially at the end of once you do your DTI analysis, you have fractional anisotropy. You have measurements of fractional anisotropy, axial diffusivity, radial diffusivity, and mean diffusivity. You have four of these entities that they give you. So, if, for example, you're looking at Alzheimer's disease, and you're looking at how a DTI in that, and you're able to find FA differences. I mean, when you do a DTI scan, you'll get um, these four indices that actually tell you, the track coherence. Axial diffusivity says whether, I mean, radial diffusivity tells you whether the state of myelin across the actual axon. So these different things tell you different aspects of diffusion imaging. Okay, I hope I'm making sense. Okay, moving on. Um,
Alan, excuse me.
Dr. Maestre, yes.
This is Gladys. Can you put back the slide? And excuse my ignorance.
No,
Uh,
no, I'll definitely do that. One second.
Thank
So,
you. So, when we see the fiber tracking and white matter delineation,
yes.
and you are using different colors, um,
Oh, yes.
to and I see in the even in the calculating of the tensors, what does that mean?
Yes, that's wonderful. Thank you for that wonderful question. Each color represents a direction. So we have now, as you know, in the brain you have superior and inferior. So if superior, inferior, so the color red means that the fibers which are going from the superior part of the brain to the inferior part of the brain. The color green talks about from the lateral to the medial, and the color blue is from the anterior portion of the brain to the posterior portion. So each of these actually talk about they are color coded to signal the direction of the particular fiber.
So when you say when I see the fiber tracking
Yes.
box, it seems
Yes.
like one arrow follow another arrow.
Oh, yes, yes, yes. Oh, let me explain that. So that is tractography. So, this is all done in um, let me step back a little. When we first started DTI many years ago in the early 2000s, we had to do it. We had to go voxel by voxel and manually kind of trace it out. Now, there are softwares that actually look at a particular voxel and look at the principal direction, and it actually marks an arrow at the principal direction. So sometimes you may have an orange voxel that shows it's kind of partially in one direction and partially in another direction, and then the water diffusion across it will move to a completely different voxel that has a different principal uh, direction of the water diffusion.
So
Uh,
like in that again third box, you have like a
yes.
red box that doesn't have any arrow.
Oh, yes, yes. So this particular picture shows that we don't choose every single voxel. We only choose certain voxels because we are just tracking how a particular vesicle goes. For example, if you're looking at the uncinate vesicles, we are going to, I mean, the software does it for you. Earlier, we used to do it like manually. So the software will take the principal diffusion direction and keep moving along with it. And this one, which you see in front of you, that doesn't have that, shows that voxel has been omitted.
Oh, okay,
Yes, we
okay,
omitted that because it's not a coherent. I mean, it's not a part of that vesicle, that vesiculus.
okay, okay. Thank you very much.
Sure.
Sorry
Thank you.
for the interruption.
No, no, no. It's fine, Dr. Maestri. Moving on. The After DTI, we also did NODDI, which is basically an advanced form of DTI. So What it means is that Noddy models diffusion according to three compartments. So you have let's just move this a little bit over here. We have, okay, we have tissue where it's intracellular, within cells, extracellular, and CSF, cerebrospinal fluid. So sorry, man, this is pretty, sorry about that. Let's go back. Okay, so intracellularly, what it does is that it models it as sticks. Oops, sorry. It models it as sticks, you know, perpendicular sticks, sticks like crisscross, and also sticks that are curved. And it's called restricted diffusion. Restricted diffusion intercellularly. And extracellularly, it does what's called as parallel diffusivity and perpendicular diffusivity. So, and then it's called restricted diffusion and hindered diffusion. And the final thing is free diffusion. So in CSF, water molecules can move anywhere. It's not restricted to a particular, it's not restricted to a particular axon. It can in a particular direction. So it can move anywhere. So what happens is that we get indices for CSF, extracellular, and intercellular. In other words, parameter maps representing neurite density and orientation dispersion index can be estimated. So, in other words, to look at the actual disease, if Alzheimer's disease, the Alzheimer pathology, is affecting the cells, we can actually look at neurite density, we can look at orientation dispersion index, and then we can compare that with people who have MCI, mild cognitive impairment, and also who are completely spared. So you can make these comparisons between people who have normal aging. I hate to use the word normal, but people who are growing old without pathology. People who have mild cognitive impairment and Alzheimer's. So you can actually use these for actually comparing how the disease is affecting uh, affecting the neurites and also the flow of water as in DTI. Okay, hope that makes sense. Let's move on. The other technique is to measure cortical thickness. And as many of you know, FreeSurfer allows you to do that. So basically, you have a T1 weighted input, which then you send in for intensity normalization, and then you do, and it automatically does it for you. It does skull stripping, it does then white matter segmentation, it does volumetric labeling, and then you have surface extraction, and then you have surface atlas registration. So it registers it to an atlas. So in other words, it's able to tell you the pulvinar is the pulvinar, the thalamus is the thalamus. So it's able to build for you an inbuilt labeled inbuilt labeled brain. Sorry, one moment. So you have gyral labeling. So it's actually, they actually kind of give you the names of it. It does it all automatically. Now, the important thing about FreeSurfer again is one needs to do a lot of checking to see if FreeSurfer is doing the thing that we ask it to do. Because at times, FreeSurfer overestimates and takes non-brain regions, or sometimes underestimates and leaves out a lot of critical regions in the brain. In other words, tiny bits of the hippocampus can be just taken away. Again, the efficacy of FreeSurfer is directly proportional to the quality of brains you give it. If you are able to give it 7 Tesla imaging brains, it does an excellent job. But if you are giving FreeSurfer a brain that is full of a very low SNR, signal to noise ratio, where you have this person in the scanner keeps, he's a mover and a shaker, he keeps moving to things. You get scans that are really bad. And FreeSurfer's estimation of gray matter, white matter, CSF, and the other forms of segmentation become completely awry. Okay, moving forward. Um, so we for that paper, we looked at functional connectivity analysis of the salience in the right frontoparietal network, and what we actually found out was that keeping the seed in the right posterior parietal cortex and looking at the target ROI as the dorsal anterior cingulate cortex, the left anterior insula, the right anterior insula, the left rostral PFC, and the right rostral PFC, we actually were able to find that um, the connectivity between the right PPC and the dorsal ACC was very significantly connected. In other words, the functional coupling between these two regions was significantly enhanced in people who smoke. So you had the right PPC. Sorry, 1 second. Yeah, the right PPC and the dorsal ACC, the right PPC and the left rostral PFC and the right rostral PFC were significantly um, the functional coupling was enhanced, and this picture actually shows you that. So you can see the right frontoparietal cortex. These red colored bands indicate enhanced connectivity or hyperconnectivity. As you can see here, you have the band between minus 4.54 and plus 4.54. So this actually shows you the maximal level of connectivity in comparison with people who don't smoke. Okay, so this is the actual output from the Con toolbox, which actually shows you the connectivity is enhanced in these regions. Okay, so moving on. Okay, so in terms of the connectivity ring, the connectivity ring shows you what the advantage of using a connectivity ring. It shows you the proximity of the connections. In other words, you can have like information superhighways from the anterior portion of the brain back to the posterior, from the orbitofrontal cortex to the occipital lobe. You have like vesicles that run. The IOFF, the inferior occipitofrontal fasciculus, runs all the way from that. It's like a information superhighway. In terms of that, we can see in that what we found out is a fairly local region, although although when I see it on the brain, you can actually see these three circles. It shows that it's not that local. It's a little bit more distributed, as you can see. So, but we still rely on the connectivity ring to estimate the distances of the functional coupling. Okay, moving on. So, what do we find out from the tobacco 7 Tesla imaging? We found out that smokers' performance on the delay discounting task was significantly lower than non-smokers, implying that smokers have greater intertemporal decision-making deficits and lower response inhibitions than non-smokers. In smokers, seed-based connectivity analysis revealed significantly enhanced functional coupling within the nodes of the right frontoparietal network, that is the right PPC and the right um, rostral prefrontal cortex and the right posterior parietal cortex with the left rostral prefrontal cortex, and between the salience network and the frontoparietal networks in smokers. The connectivity strength between salience network and frontoparietal network predicted scores on the delayed discounting task in non-smokers. What this means is that the greater the coupling strength, the better the score on the delay discounting test. Okay, moving on. Tobacco cigarettes did not significantly affect brain structure. So brain structure was basically intact. It did not affect connectivity or microstructural architecture. So tobacco cigarettes only affected cognition and only affected functional connectivity. The other structural connectivity and the individual volumes were spared. Now, I'm not terribly sure about that because when it's because FSL DTI does something which is very kind of irritating in that it does a multiple comparison correction for the whole brain. When you do multiple correction for the whole brain, very tiny, very significant findings are just washed away. You have so many of these interesting findings just washed away because multiple comparisons corrections is the key to everything. I mean, otherwise it's just a fishing expedition. So I did see when in the uncorrected scans that there was so many tobacco is affecting a lot of like white matter structures. But once you apply Bonferroni, which is other in the other word, other meaning of multiple comparisons correction, many of these findings are just completely washed away. But that is how you can publish. Otherwise, without Bonferroni, you just can't publish. So I just need to kind of sort of throw it out there. Anyway, moving on to cannabis at at 7 Tesla. One of our earliest papers gave us an indication as to what to kind of look for, and this is with my group. So one of my colleagues at Massachusetts General Hospital, and we actually sort of published this, and that gave us a lot of that opened up lots of interesting questions as to what to look for in 7 Tesla imaging. So one of the things we actually looked for in Tesla, which was actually recently published. No, actually it is published in 2023. We actually were able to find out that there was functional hyperconnectivity between corticocerebellar networks. So we found that there's a lot of cerebellar, um, you know, involvement um, in the activity of when people smoke when people smoke cannabis, and that was involved with altered decision making in young adult cannabis users. So in for this particular study, our study design was we used um, around we used 90 cannabis users and 90 non-users, and we asked the principal the question we asked was, does smoking cannabis affect functional connectivity of the cerebellum? Just a moment. The second thing that we asked was, does smoking cannabis affect decision making as as looked at by the Iowa gambling task, which is again a standardized task. And then we look, one of the other questions we asked was, is there a relationship between cerebellar functional connectivity and decision making in cannabis users? One second, let me just have a swig. So we did seed-based functional connectivity analysis of the anterior cerebellum, and we found that the left anterior cerebellum was significantly um, it showed significantly increased connectivity with the left pallidum, the left putamen, and the right nucleus accumbens, as you can see there. And this was a family-wise error-corrected analysis. So in terms of the actual picture, this is what it looks like. So you can see here the anterior cerebellum. The cerebellum is over here. The anterior cerebellum is functionally connected. The color red, sorry, the color red represents increased connectivity with those three regions, the pallidum, the putamen, and the nucleus accumbens, okay? And then we looked, so if you look at it from a transaxial view, you can actually see this. So this particular thing is the anterior cerebellum, and you can see the putamen, the pallidum, and the nucleus accumbens. It's higher connectivity in this, in people who smoke cannabis. And then we looked at seed-based connectivity of the posterior cerebellum, And we found that the posterior cerebellum, one second, is again has increased connectivity with the default mode prefrontal regions, the default mode right lateral parietal regions, the regions that are close to the salience network, which is the left and right um, the left and right paracingulate cortex and the subcallosal cortex, and all of them were very highly significantly. Um, in other words, the functional coupling between the posterior cerebellum and all of these different regions was very highly significant. So as you can see in this picture, you have the a seed region as the posterior cerebellum, and you have the lateral parietal here, and the left and right paracingulate, and the default mode networks, two seats, the subcallosal cortex and the medial prefrontal cortex. I think that's what I was looking at. Just a second. Yeah, default mode, medial prefrontal cortex. So we have both now. We have the anterior cerebellum involved and the posterior cerebellum involved. Now let's bring them both together. Okay, looking at it from a transaxial view, we can actually see the posterior cerebellum over here. Sorry, goodness, yeah, the posterior cerebellum here, and the way it has been connected to the left and right. This is the medial prefrontal, the lateral parietal. This one is yeah, this one is the paracingulate, so you can actually see those connectivities over here. Now, bringing them both together, we have what is called a cerebellar hyperconnectivity. So you have anterior cerebellum and posterior cerebellum hyperconnected to many of these regions in the brain. So to put it in perspective, smoking cannabis in young adults causes hyperconnectivity between two cerebellar networks, the anterior cerebellum and the posterior cerebellum, with several regions in the anterior portion of the brain. So you can see the posterior cerebellum and the anterior cerebellum. And looking at it from a transaxial view, this is the picture that emerges. Okay. Now, in addition to our functional connectivity findings, we did something called as a data-driven AI method called whole brain multivariate pattern analysis. This is an omnibus test of connectivity. Okay, so you're saying there's so much connectivity. Let's do an omnibus test and see if we really have what we do. And then and this was also FDR corrected, which is a family-wise error correction technique. And what we actually we actually found out was, just a second, let me just move this a little so you can see that. It's not moving. Okay, here you go. Okay. What we actually found out was, just a second. Okay. Remember we talked about the connectivity ring. The connectivity ring is the actual structural basis. So we can actually see the cerebellum here. Cerebellum in this picture is strongly connected to several regions of the brain, including the anterior cingulate over here and the precuneus. Many of these regions are part of the default mode network. You have the salience network here. You have language areas over here. So the MVPA of 7 corrections with 175 subjects gave us a 4.98 uncorrected and a p-value FDR corrected of 0.006. So in other words, what it was saying to us is that there is indeed this hyperconnectivity in cannabis users. Is it making sense? Thank you. Let's move on. So what do we conclude from cannabis? Multimodal 70 imaging data shows that cannabis affects the the dynamic brain, while the static brain, just structure is relatively preserved in early stages. So cannabis affects the way cognition is done. Cannabis affects functional connectivity, but again, you don't have concomitant structural problems. Cannabis users' performance on the Iowa gambling test was significantly lower than non-users, implying that cannabis users have greater decision-making deficits and lower response inhibition than non-users. The anterior and posterior cerebellum show enhanced connectivity with regions mediating both addiction and salience. The addiction was a region called the nucleus accumbens. It anterior posterior cerebellum are connected to the nucleus accumbens, which is a key region for addiction. It's called the reward pathway. So nucleus accumbens sits right in the middle of the reward pathway. So both of these actually sort of connect with the regions mediating addiction and salience. The MVPA confirmed the functional hyperconnectivity of the cerebellum in cannabis users. The cortical volume of the left nucleus accumbens was significantly larger in cannabis users. Okay, yeah, yeah, that was as far as the cannabis at 70. We then looked at, is it different between women and men? We looked at sexual dimorphism in cannabis users, and we actually published this paper fairly recently. It's coming out in January 2025. These sex differences in intertemporal decision making and cortical thickness of the orbitofrontal and insular in young adult cannabis users. Evidence from 1,111 subjects. So we had a huge kind of sample, and I did it with my collaborator at Harvard University and Dr. Francesca Philby, who was at UT Dallas, and she has actually sent me her cannabis data, which is of excellent quality, because she has herself taken care of a lot of most important aspects of this whole addiction thing. So I'm kind of looking forward to analyzing that as well. What we actually found out from this is that young adult cannabis users had thinner cortices in the bilateral insular and orbitofrontal regions. Now, what about sex differences? Yes, there were sex differences. Male users had significantly thinner bilateral OFC and insula, whereas female users had only significantly thinner bilateral OFC. Their insulas were spared. The orbitofrontal cortex is involved in reward and decision making, as shown by Edmund Rolls in 2021. Hence, thinner cortices may imply deficits, and I say may imply because this is, it's you can't say it for absolute certainty. It may imply deficits in motivational, affective, and emotional processing in the pursuit of reward. Female users' bilateral insula did not show cortical thinning. It was absolutely normal. And this is for, I mean, we were looking at something like 600 young ladies, which means that interoceptive awareness, that is, the knowledge, interoception means the awareness of oneself. For example, when you're standing in a room, you're aware of your heartbeat. You can actually hear your heart beating. That is an interoceptive awareness. Interoception and salience go hand in hand. So when you're walking fast and your heart is beating hard, you're aware of the fact that, oh my God, my heart is beating too fast. I need to decrease my speed. All of these it's very important. And I want to tell you that NIH is very interested in interoception. That is the connection between the brain and the heart, the heart-mind connection. So in other words, females users bilateral insula did not show cortical thinning, which means that interoceptive awareness, which include thoughts on craving, was in the was in the normal range. So they didn't have that particular problem. Okay, moving on. Let's look at alcohol at 7 Tesla. So after I came to UTRGV, we did a major alcohol study, and Dr. Saloom was also a part of it. We we looked at brain functional connectivity patterns in young adults across the alcohol dimension. Now, the alcohol dimension is the whole panoply, the whole looking at no alcohol, mid alcohol, and high alcohol. So you're looking at the whole dimension. So very few studies look at the entire dimension. They either look at alcohol or no alcohol. That's heavy alcohol, no alcohol. Now we looked at the actual dimension and looked at functional connectivity in that. So in this study, we had 69 subjects, 23 heavy alcohol, 23 moderate alcohol, and 23 no alcohol. Again, we applied family-wise error correction and false discovery rate corrections. 3 comparisons were made. We looked at heavy versus moderate, heavy versus no alcohol, and moderate versus no alcohol. Across all 3 comparisons, alcohol using subjects showed increased functional connectivity between and within networks. The greatest magnitude of functional coupling and the number of regions correlated were between heavy alcohol and moderate alcohol. We again did a data-driven multivariate pattern analysis that showed significant hyperconnectivity differences only between heavy alcohol and moderate alcohol users. So the MVPA gave us this image, and the connectivity between these different regions, these connectivity bars shows increased connectivity. If it was lower connectivity, it will be in blue. So we actually have as you can see, we have both salience network, salience ACC standing for anterior cingulate cortex, and also anterior cingulate. Now this little guy, this little purple picture, this circular picture is the salience ACC. The AC stands for this big guy. So when you see both AC and salience ACC, they're not the very same things. Salience ACC is a part of the anterior cingulate cortex. It's a little bit confusing, but that's the way the Con toolbox generates it. So the functional connectivity differences between heavy alcohol use and light alcohol use is given here. So we found hyperconnectivity between the salience ACC and language areas. That's left triangularis and left opercularis. We also found the left supramarginal gyrus, left middle temporal gyrus. We also found hyperconnectivity between the left putamen, the left pallidum, and language areas, as you can see in the picture. We also found right putamen and right pallidum, and left supramarginal gyrus, and left middle temporal gyrus. All of these are hyperconnections. And we also found hypoconnectivity, which is lower connectivity, was observed between the orbitofrontal cortex and the left language areas. Now, to put it in perspective, these connectivity bands actually explain the level of connectivity. Now, when you say connectivity, what the heck does that mean? It means the rate of oscillations from a given group of neurons. The rate of oscillations from a given group of neurons is temporally synchronous to the rate of oscillations of a different group of neurons. So for example, if you see 4.37 here, it's a higher rate of oscillation. And this is temporally synchronous with 4.37 in the inferior frontal gyrus opercularis. Okay, so these the oscillations from a given group of neurons happens in temporal synchrony. If it is temporally asynchronous, they are anti-correlated. Am I making sense? Okay. So functional connectivity means a group of neurons activating together. In other words, yes, activating together, that is oscillating together. Okay. So the actual this map which I showed you, I'm just sort of explaining that with a different map. So this is not different from the other one. It's just an explanation of the previous map. Okay, and if you look at it in the connectome ring, you can actually see the degrees of connectivity. So red is the very strong connectivity. Um, Orange is a little bit less, and brown is not that much, whereas blue is hypo connectivity. So you can actually see the levels of connectivity between two different regions. So then, when you submit a paper, then you can actually, all of these explains to a reviewer, what the heck is happening. That is, you know what what is this connectivity kind of telling us. Now, interestingly, with these different bands, our next step, what we're going to do is called connectome harmonics. So connectome harmonics is the very next step. We're looking at how these oscillations are not quantitatively different, but qualitatively different. Now, we've already established there's a quantitative difference between them, but now we're going to establish, are they qualitatively different? Harmonics comes from the fact that, when you play an instrument, you actually see the different harmonics. So, I play a musical instrument, so I'm very interested in knowing how the connectome harmonics are different between different regions, looking at how there is a qualitative difference between different regions that are functionally connected. Okay, moving on. Okay, one thing, we have submitted this paper. It's now in the review process. They sent it back. They wanted a lot of changes. They wanted a lot of explanations. I explained all of it. It's gone back. Hopefully, it'll get published. Now, we looked at diffusion tensor imaging in young alcohol users, and this time I dropped FSL DTI and I used Dracula. Dracula is a different beast. You cannot do Dracula on an ordinary computer. You need to do it on a computing cluster. A computing cluster is a group of. It's a huge. It's a whole network of computers. It's like 1,000 computing units linked together. So essentially, what you would do is you'd send a single brain, and at the second step, it's called the bedpost X. It runs it all in parallel. It runs it, so it's like 100 processing in parallel. It's really hard. When I was at MGH, we used to actually have a lot of competition to send these things in. And when I came down to the Rio Grande Valley, I was thinking, how am I going to run? How am I going to run? How am I going to run Dracula? Um, you know, um, Luckily for us, we've been able to get these computing systems, especially a computer called the Mac Pro. It's not the MacBook Pro, it's the Mac Pro. The MacBook Pro cannot do it. The MacBook Pro would just break. If you use a Mac Pro, it's like a mean beast. It can do it. They have GPUs there, graphical processing units, and they also have like 192 I'm sorry, 192 gigabytes of RAM. So you can actually do it. It can actually simulate what a computing cluster can do. And I was so happy when we could actually do this here, because I was hoping I could convince Dean Hawker to get a computing cluster, and I don't know where that'll go. Anyway, the important thing is we managed to do that, and we ran Dracula. And this is again a paper that we're actually working on. We're going to actually, Dr. Salom and I are going to kind of publish this we first presenting it at a conference, and hopefully once that is over, we can actually publish this in something else. So what we actually found was that, one second, and okay. Alcohol use significantly lowers the middle cerebellar peduncle FA. One second. Yeah. So if you look at the cerebellum, this little blue region is called the middle cerebellar peduncle. Alcohol use significantly affects the FA, the fractional anisotropy in. So if you have alcohol, this is what happens. There's an FA problem there, which then leads to difficulties in motor coordination It also leads to cognitive deficits that which are because we've now found out that the cerebellum is involved in cognitive function as well, not just not just in like motor function. Okay, so okay, so that's as far as 5 papers are concerned. I don't want to keep going on with it. I can, but I don't want to. What do we understand from nicotine, cannabis, and alcohol? Okay, what are the nuggets of wisdom that we get out of it? Consumption of these three substances individually and together cause hyperfunctional hyperconnectivity between several regions of the brain. It also causes decreases in fractional anisotropy and other DTI modalities, as shown by our studies and other studies. It's not just me who's actually showing it. There are other studies that have shown it. In general, cortical thinning in brain regions associated cognition and behavior are there. We also have altered cognitive function in decision-making abilities. That is across the entire board. These two cognitive functions have problems. You have problems in impulsivity, problems in decision-making. There is sexual dimorphism in this, but the exact nature across all three are not very clear. We have understood some things, but we still have like a, a, a lot of ways to go before we actually do it. The microstructural architecture using NORI appears to be relatively spared in all three substances. Okay. What causes functional hyperconnectivity? Functional hyperconnectivity is caused by compensatory mechanisms. Hyperconnectivity as a compensatory mechanism to counterbalance structural damage. So if one person takes alcohol, alcohol is H2H5OH, it's ethanol. It causes damage to the brain. The molecule of H2H5OH, when it enters across the blood-brain barrier, it does structural damage. Now, what does a person's brain do? It compensates by hyperconnectivity. Likewise, when you smoke tobacco, you don't just inhale nicotine alone. Along with nicotine, there are so many substances, including benzopyrene, which is tar. Tar causes a lot of damage to both the lungs, and it also causes damage to a person's brain. So all of that could cause compensatory um, mechanisms, one of which is hyperconnectivity. It could be altered network function. Drug use can disrupt the brain's normal connectivity patterns, particularly in networks like the default mode network, salience network, and executive control network. Functional connectivity may reflect a, maladaptive reorganization of brain activity. So when a person smokes a cigarette or smokes a joint of cannabis or drinks alcohol, there is a reorganization of activity in the brain. When it reorganizes, it contributes to cognitive function, emotion regulation issues. For example, people who drink alcohol regularly get abusive, they get very angry. So that's all a part of the emotion regulation problems and impulse control. Functional hyperconnectivity could also be because of impaired inhibitory control, failure of the normal inhibitory processes. In drug-dependent individuals, brain areas that should be relatively inactive during rest, such as your prefrontal, especially the dorsolateral prefrontal, which is responsible for self-control and also judgment, may suddenly be overly active, which could relate to an increased craving and lack of impulse control. And then functional hyperconnectivity increases the risk for relapse. And this is the key. Increased connectivity between reward-related regions, like the nucleus accumbens, which we showed, and the prefrontal cortex, might be associated with a heightened sensitivity to drug-related cues, which could increase the risk of relapse in recovering individuals. Okay. Okay, what are we doing about this? Okay, we have all these issues. The Rio Grande Valley has all of these different issues with alcohol, cannabis, and smoking, one of the things that we do at the Institute of Neuroscience is high definition transcranial electric stimulation, which is when we put a network of electrodes on a youngster, and we are able to gently sort of give them an electric stimulus that improves connectivity in some regions and decreases membrane potentials in a different region. So let me state that again. Transcranial electrical stimulation basically works on incremental neuroplastic changes. We are making tiny changes that allow the person to make informed judgments. Okay, it could be the dorsolateral prefrontal cortex that you are stimulating. It could be the insular cortex which you are destimulating. In other words, you are stopping the insular cortex from pushing the person towards an addictive substance. Okay, so we have both the electrode, the anode, and the cathode. So you have as an inward in the anode you have an inward current direction. In the cathode you have an outward current direction. At the cathode level, you decrease a person's function. At the anode, we are increasing the person's connection. Okay, so that's kind of work that we do at my lab, and Dr. Saloom has a transcranial magnetic stimulation lab. In 2025, we're going to get transcranial focused ultrasound stimulation. That's we in my lab we're going to have that. Basically, focused ultrasound is able to enter into regions that electrical stimulation cannot reach. It can go deep brain stimulation, being non-invasive. Um, Earlier, deep brain stimulation means you had to open up the brain and stimulate. You should put an electrode, right? And using stereotactic surgery, you stimulate the basal ganglia in Parkinson's disease. But here, you can actually go to the basal ganglia. Remember the putamen and the pallidum? You can actually stimulate them using a non-invasive technique called as transcranial focused ultrasound. This is what we are actually doing at the moment. So with these words, I thank you for your attention, and if anyone has any questions, I'll be happy to actually take them.
Thank you very much, Dr. Francis. This is just amazing.
Thank you.
I still needs to, and again, excuse my ignorance, but when you were explaining the connectivity, you basically said that there are two different areas that are vibrating, or correspondingly, and
Yes.
you show this. Now, it is not clear to me how you define hyperconnectivity.
Okay. Okay. So, um, I mean, so, um, hyperconnectivity is if we look at the connectivity across a sort of group A, and we look at the connectivity in like group B, if the degree of connectivity is significantly higher in one group, the degree of connectivity, that is the degree of, when I say connectivity, it's functional coupling. They couple together. The functional coupling is significantly higher than the other group.
You mean the other experimental group, not the other group of neurons?
Oh, so um, yeah. So I mean, so I was talking about smokers versus non-smokers.
Okay.
Let me just look at smokers versus non-smokers. So when we're looking at the salience network and the frontoparietal
Mm-hmm.
network, what we actually find is that the degree of coupling is significantly higher for people who actually smoke. The degree of coupling. So And that is a mathematical calculation that is done um, by the software. So um, so what I what I actually mean is that it's a higher rate of coupling, a significantly higher rate of coupling in comparison with someone who really doesn't smoke or drink or any of that.
So it's like not all the neurons are um, oscillating in the same way.
Correct. It's just when you're looking at a particular network, say, look at the actual salience network, which is a group of neurons is constant, either the insular or the anterior cingulate. So um, when they oscillate together with the frontoparietal network, they oscillate the oscillations between them in this group is significantly higher than the oscillations of the same network in a different group. That's how it is.
So it might be the same magnitude, but more neurons, is that?
Um, That's an excellent. It could be more neurons, but more synapses, more receptors, more a number of receptors that are undertaking these oscillations. So the understanding is that drug addiction causes the increase of receptors in certain regions in the brain, and that is an inbuilt mechanism to drive a person towards an addictive habit.
Mm. Thank you so much. Very interesting.
Yeah. I think I can clarify a little bit about that too. I worked on that field too some time ago. The thing is that hyperconnectivity doesn't mean that it's connected better or more. It's the high correlation between the activity in one place and the other place. And
Yes.
this is a disturbance actually. It happens in children with autism, and some of the mental disorders. Many mental disorders have a lot of hyperconnectivity in this sense. So an advantage. It's a problem actually. So
Yes.
people having yeah, I had here for example a major depression. It happens especially in children. Autism in preterm children, they also have a lot of hyperconnectivity. So it's a dysregulation of the activity between areas that is happening there. That's basically what is the increased connectivity, the hyperconnectivity related to. Yeah. I see Alejandro Lopez is having a raised hand. Thank you for joining, Alejandro.
Hello, everybody.
Hi.
Nice talk. I'm
Thank you.
always curious about these techniques of in vivo imaging and how the
Uh-huh.
brain changes with experience. So obviously, in this case, we're not talking about changes of neurons in the brain because it
Right.
would these things are mostly learned when you're adult, right?
Right.
Ideally, if people start smoking, drinking, everything already when they are mostly adult, the brain. And so, and just making that clear first, I would like to ask that
Yes.
what the postmortem studies say about the cellular or even molecular changes in terms of these two techniques. First, functional MRI, and
Mhm.
second, DTI MRI. So functional, yes, could be telling us about the let's say changes in activity, more receptor, more synapses, synchronity, and many things, right? But
Mhm.
what changes, and if that correlates with the physical, with the things that the DTI MRI is telling us, because it's nothing related to functionality, it's
Right.
just structure. So what is the correlation? And second, what will be the cellular explanation for this learning in addictions? Because everything is learning, even addictions.
So thank you for that. With like people who are dead, that is people who for postmortem, we can do structural imaging, absolutely, and we can do diffusion tensor imaging, but we cannot do for doing functional connectivity. there is no activity in the brain.
No.
Yeah.
Postmortem, I mean, when the information about how these people had their hyper connectivity and everything, what
Yes.
the cellular correlate is what
Oh, the
happened
cellular correlate.
else.
So I mean, from uh, I mean, from people who are opioid addicts, I can give you that. I'm not 100% sure about the PM postmortem imaging in nicotine and others, but with opioid addicts, what they really find is an increased receptor density. At the cellular level, what we find is there are more receptors. Um, uh, the mu opioid receptors, the kappa opioid receptors, have have been increased dramatically. So essentially, when a person has an opioid, either as like heroin or any of those things, these receptors actually drive the addictive process. So what they actually have found in people who are no longer living is that there's a higher level of receptor density.
And that would be changing the DTI MRI.
Um, well, when you look at it in people who are alive, yes. When in people who are alive, people when you smoke cannabis or any other thing, there is a change in the DTI. That is also observed at the cellular level when people are no more. So you can actually see, you know, sort of reductions in FAA and other things in the in the postmodern brains.
Okay, cool. Thank you very much.
Thank you.
Megan, before we get to your question, there was a question in the chat box by Jenna Zamora, which is, did long-term or higher amounts of usage affect nicotine, alcohol, cannabis users more? And did the route of consumption affect anything as well? And you can
Um,
see then you can see the question again in the
okay.
chat box as well.
Just let me just quickly just look at it. Or Mera, can you explain again? I didn't get it.
Yes.
Uh,
So the question is, did long-term or higher amounts of usage affect the users more? And
um,
did the route of consumption affect anything else as well?
that's both of these are excellent, um, excellent questions. Um, we didn't look at long-term use, but looking at the addiction literature, yes, long-term use affects the brain profoundly, and long-term use also makes it very hard for people to sort of stop the addictive habit. S- That is one. Second, the route of use. Yes, if you take any sort of drug, like sort of intravenously, it's going to be more potent. I wouldn't use the word effective, but more potent, and it would sort of hit the regions in the brain um, much more efficaciously than if you were to just sort of smoke cannabis as a joint,
Mm.
so.
Thank you so much for that. Megan, your turn.
Hi, so I really, really loved the presentation. I thought it was a wonderful way to really demonstrate just the full range of abilities that these different imaging processors can do and these different databases. I was wondering if we could go back to the diffusion tensor imaging, if you could explain the FA, MD, AD, like those things a little bit more, please.
Absolutely, let's do that. Oh, by the way, this Megan Francis is my daughter. So anyway, let me show you that. Let's go to that. Okay. Let's see. Play from the current slide. Okay. Okay, okay, one moment. What did you want to know?
Or what FAND, what those standard for, and like
Okay.
how, just a little bit about how they connect with one another.
Okay, absolutely. When you take a drop of water and you put it on a table, it moves in every single direction. That's a typical physical phenomenon called Brownian motion. Now, when water diffuses across the axons, um, uh, essentially that the rate of diffusion can be actually calculated. Now, fractional anisotropy is a way of looking at how water diffuses across axons. So, if in a person who doesn't take any addictive substance, if you look at the FA in that person, the the actual rate of movement of the water across the axons will be similar to everybody else. Um, so when you compare this with people who have an addictive habit, um, what happens is that the addiction causes them to have lower FA. You know, so it's actually a, a, a sort of a way of calculating how the white matter tracks are actually coherent. It's track coherence. Am I making sense to you?
Yes, that makes sense.
Okay, so yeah. So when you actually look at the FA, you also look at how this track is it going straight or is it curving, and then you go into something called as fiber tracking. So you actually see where it's going by looking at the principal diffusion coefficient. It actually shows you how it goes. But in earlier days, we used to actually do it voxel by voxel and see how the principal diffusion coefficient goes. But in today's world, the software will actually do it for you. But you need to know what this implies. Am I making sense to you?
So for the bigger picture, how do you derive a connectome from this imaging? Like how does the levels build up to get to that point that like the Con toolbox can show you?
Yeah, excellent cue. Um, So, what DTI will actually allow us to do is called the structural connectome. So it actually tells us what, if you look at this is basically a structural connectome. It tells you where all the different fibers lead to. And if there is a problem in the fibers, in other words, if there is a track coherence problem, you can actually see it in that. So DTI gives you the structural connectome. The Con toolbox allows you to have the functional connectome because it does functional connectivity studies.
I see, that makes sense. Thank you.
Sure. Any other questions?
Oh, it's a very nice talk, Alan. Really
Thank you.
enjoy your presentation, all the studies and all this stuff. Is I think is very important for the advance of this field. Something I would like to put just a small question is what besides the evident advantage in resolution and improvement in the images. I see that the images from the 7 Tesla, they are higher resolution in one of on the two sides of the boxer, but the other side keeps being the same size as the usual
Yes.
3 Tesla. This may be the carriage inside
Yes,
the device, no?
yes, yes, yes.
Which makes sense to do
Yes.
something with that. What,
Yes.
why they haven't improved that part of the resolution, that
Yes.
dimension?
Yes. First of all, thank you for that particular question. So, I didn't tell you the actual drawbacks of 7 Tesla imaging. First of all, 7 Tesla imaging means that it's a bigger magnetic field, and sometimes patients cannot go in, especially if you have a tooth implant, or if you have an implant in your brain, or any, it's, I mean, you just can't go in basically, and you have to declare that. Secondly, because of a higher signal to noise ratio, if you move a little bit in the scanner, the image is all gone. You got to really stay still. So because of that, they put like a pillow and they just totally kind of box you in, and it's not very comfortable if you're doing like a 1 hour scan. It's even for people who are young, who are healthy, have a lot of difficulty just standing, just being still. And if you're doing like activity, like, if you're doing like functional scans where you have to respond to things, if you move, again, they have to do a completely other thing. Now, coming to your thing, why is the image not that big? It can be made bigger, but what they're, I mean, the basically using 7 Tesla with a 64 channel coil optimizes the molecular movement, so that the movement up and down, so you get this, so you're able to get a very clear image. So that's as far as I can put it.
Yeah, no, that's great. Well, anybody has any more comments? I think we reached over the time, but definitely we enjoyed this talk with you, Alan. Thank
Thank
you.
you.
Thank you
Thank you.
again. Thanks for presenting all these advances. I think it is very important. And well, thank you for everybody who joined us today here. We had a very nice crowd today.
Thank
You
you.
again. And thank you, Nira. Well, we have to say thank you, everybody, and also Happy New Year and
Happy New Year.
Merry Christmas and all the
Merry
holidays
Christmas.
that are coming in this time for each one of you. And we will see each other again on the Tuesday, January 14th, who's going to be presented by Andrea Mendez Colmenares for the next seminar. Thank you so much.
Excellent.
Thank you, Neira,
Thank you
again
so
for all
much.
the
Thank you so much,
coordination.
everybody.
Thank you so much.
Thank you.
Hope to see y'all in
Well,
2025.
thank you.
Bye-bye.
Bye.